All right, ag biology. Today we're going to move on and continue some practice with protein synthesis and decoding those codons. Yesterday you did a really basic worksheet that was just to help you read those codons, 3 nitrogen-containing bases at a time, determine which base pairing rule you need to use where. Remember, apple in the tree, carrot in the ground with DNA. And then keep in mind that with RNA, we don't have any thymine, it's all uracil. So we have apple under, and then carrot in the ground remains the same. Today you're going to do another worksheet that's very similar. I just want to go over the directions with you to make sure that you're clear on everything that you're going to do here. So as you're working through this, you'll see here I provided you with a DNA strand. Your first job is to use the base pairing rule to figure out the messenger RNA. Okay, pretty straightforward. So if you have a thymine here, what are you going to put here? That's right, adenine. Okay, cytosine, guanine. You can figure out the rest. Okay, that's that complementary messenger RNA strand. Okay, that's that monk opening that recipe book, copying down the directions, throwing this one out the window. Okay, now keep in mind that this strand gets thrown out the window and it lands on that big fat ribosome. And now these guys, these transfer molecules, are going to read the messenger RNA 3 nitrogen-containing bases at a time and bring along with them an amino acid. Okay, so go ahead and use the base pairing rule to pair tRNA with messenger RNA. Keep in mind these are both RNAs, so there's going to be no thymine. It is going to be uracil only. Once you've done that, you need to figure out the amino acids. This is where a lot of people stumble. Keep in mind that amino acids, when you're using your codon chart, okay, and you can use the codon chart from the PowerPoint yesterday, so just open my PowerPoint from the notes yesterday and use that, or you can use the assignment that you did yesterday that was passed back to you. Okay, when you're reading messenger RNA or transfer RNA to get yourself an amino acid, you actually read this one, okay? So the transfer molecules just base pair with them, so it's like fitting the puzzle piece or putting a key in a lock. The messenger RNA is the one that codes for this. So as you're using your codon chart, make sure you're using the messenger RNA to get your correct amino acid. Okay, that should be pretty straightforward. The next set of questions is going to require you to circle one. So you're going to see a lot of these questions. Messenger RNA is made during, and then you need to circle transcription or translation. Use your notes for this. I also encourage you to use your textbook to get further explanation. Okay, and then you're going to do the same concept here. You're going to go through and fill out the messenger RNA, tRNA, and determine amino acids. And again, go through some questions. Same thing here. Okay, so these questions that have all these parentheses in them, it's wanting you to circle the correct answer. Okay, so I just wanted to clarify that. You should be able to jam through these pretty quickly. If you don't want to take the time to pull up my notes from yesterday, you can utilize this codon chart down here at the bottom. I just like the way the other one looks better, but you're more than welcome to use this one. Okay, so if we're reading our messenger RNA, you're going to start on this side with your first nitrogen-containing base. So let's say it's A. That means you're going to stay in this row. And go to your second nitrogen-containing base. Let's say it's C. So we're going to be in this box somewhere because we've got A, C, and then for your third letter, you would just come over here and slide over, okay? So that would be A, C, U, and then this right here is going to be, excuse me, on this side, whoops, this is going to be your amino acid. That's why I don't like this chart very much. Use this one or my circular one, whichever you prefer. You should be able to jam through this in 55 minutes. Let me know how far you get and we'll determine what we're going to do the next couple days, but I don't see it being a problem. You've got some stuff here that you're going to need to do some research on, so please use your textbooks. You can use the internet too, but remember it's not as reliable because I use the textbooks. Okay, so go ahead and do your research. Use your class time wisely. Remember to be kind and let me know if you have questions. Thank you.